Hello, everyone. Welcome back to my video tutorial for spatial dataset analysis. Today, I'm going to show you how to analyze the Nanostring human pancreas dataset. You can use this link to download the data. Once you download the data and unzip the files, you will get data like I have in this folder. You can see we have the CSV files for the expression matrix data, field view, position files, metadata, TX files, and the polygons. We also have the folders to save the cell composition, cell labels, cell overlay, and the compartment labels. When you download the data, you also have a HTML README file. If you read the README file, it can help you understand the data we have here. So now we can load the data into R to do the analysis. So we can go to R. You can see we are going to use thread and analyze the data set. So the files I showed you, they are in this folder. We are going to use the load nanostring function to create a thread object. We can name the field of view as Pancreas, so C will be nanostring. So let's load the data to create a thread object. So we name it as nano object. So it will take some time to load the data. Okay, you can see we loaded the data and created the thread object. So we can click the thread object and have a look at the data. So you can see in this data set we have 21,731 genes and more than 48,000 cells. You can see in the C we have the nanostring C, and also we have the image information in the thread object. So now we can analyze the cells using the thread workflow. So we are going to use the C transform method to normalize the data, find the variable features, and scale the data. Let's run its C transform function first. Okay, after its C transform, we can run PCA. After PCA, we can run UMap. Now we can find the neighbors and also find the clusters. I'm using the resolution 0.3. You can increase the resolution to get more cell clusters. So we finish the throughout standard workflow to cluster the cells. Now we can use the plot to look at the cell clusters if we run. You can see we generated the UMap because I'm using low resolution for the demonstration. So we only have 8 cell clusters here. Then you can use the feature plot to look at the gene expression in each cell clusters. For example, we can use the GCG, insulin, somatostatin, and PPY to look at the endocrine cells. If we run the feature plot for those marked genes, we run it first. So you can see from the feature plot the gene expression. Cells in cluster 5 are endocrine cells. I run the other marked genes, cluster 0, 2, and 4. They are axilla cells, 1 and 7 are ductal cells, and 3 are immune cells, mesenchymal cells, 6 are endothelial cells. So you can run other marker genes to name the cell clusters. I'm not going to show you how to name the cell clusters today. You can watch my other videos. So we cluster the cells now. We have 8 cell clusters. Because this is a spatial data set, we have the image information when we create the threat object. So we can use the image dim plot function to look at the cell clusters on the image. We set the field view as the pancreas. So now we can run the image dim plot function to see the cell clusters on the tissue. You can see we generated the image and the cells are colored by their color code from 0 to 7. Because this data set has 2 areas analyzed by the Nanostring CosMx technology. So you can see 2 images here. So you know the pancreas is the organ that produces insulin. So we can have a look at the insulin expression cells. They should be beta cells. We can use the image feature plot function to look at the features of the insulin. Let's run. So insulin expression, beta cells are in red color because they have a high level of insulin expression. So now you can see the red color on the tissue. You can see insulin expression cells formed the islet in the pancreas. Here you only see insulin expression cells. We can also use the image dim plot function to look at the molecule of the insulin. Let's run the image dim plot function first. You can see now the difference. The insulin expression cells, beta cells, are in red color, but at the same time, we label the other cell clusters from cluster 0 to cluster 7 according to their color code. So that's my demonstration. I only use the insulin as the example. If you are interested in other cell types such as alpha cells, delta cells, or axilla cells, or even immune cells, you can use the image feature plot function and the image dim plot function to look at other gene expression on the tissue. So I'm going to stop from here for today's demonstration. I hope my video tutorial can help your data analysis, and see you next time.